In this video, we will begin to study viruses. We start by examining the characteristics of viruses and how viruses are classified. Topics covered in this lecture include the genetic material of viruses, hosts of viruses, and sizes of viruses. In Unit 1, we learned that microorganisms were first observed in the late 1600s and could be observed using light microscopy. Viruses are much smaller and could not be observed with a light microscope and therefore would continue to remain a puzzle until the late 1800s. In fact, the term virus in Latin means poison. This term was proposed by Pasteur because while scientists could not observe them, they knew that something smaller than bacteria existed that could cause disease. Viruses are very different from the microorganisms we have studied this semester. Viruses are acellular entities that are infectious. Acellular meaning that they do not have cell structure. They are obligate intracellular parasites because they require a host cell's machinery and metabolism in order to multiply. Most people are terrified of viruses, largely due to the fact that they are associated with disease. In fact, viruses do cause diseases in organisms, and most of the infectious diseases of industrialized nations are viral in nature. But not all viruses are harmful to humans. A group of viruses we will study, known as the bacteriophages, have been very useful in the lab setting. One useful application of the bacteriophage in the lab setting is the phage typing that was explored in Chapter 4 that is utilized to identify bacterial species. Other viruses, referred to as harmless viruses, have been utilized experimentally in trials to treat genetic diseases like cystic fibrosis and sickle cell anemia. The normal gene is inserted into the virus, and the virus is used as a vector to deliver the gene to the patient. Viruses do not exhibit all of the characteristics of life, and their structural organization is very simplistic in comparison to cellular structure. Viruses lack a cell membrane, cytoplasm, and organelles, including the machinery required to synthesize proteins. Their lack of organelles also prevents them from carrying out any metabolic pathway. Viruses cannot reproduce independently, but rather utilize the organelles and metabolic pathways of their host to replicate and produce more virus particles. Because of their characteristics, many scientists do not consider them to be living organisms. We will examine them as non-living chemical entities that have an extracellular state and an intracellular state. The virus exists in an extracellular state composed of a nucleic acid and a protein coat known as a capsid. The combination of the nucleic acid and capsid are referred to as the nucleocapsid. Some viruses will have a phospholipid envelope that is derived from the host cell membrane. Those with an envelope present are called envelope viruses, and those lacking envelopes are referred to as naked viruses. The outermost layer of any virus particle acts to provide protection of the genetic material and holds the proteins that will interact with host cells. An intact virus capable of being infectious is properly called a virion. After a virus has gained access to a host cell, the protective coat is removed and the nucleic acid is released. The nucleic acid is only able to redirect the host cell's metabolism if it has been freed from the capsid. As we have seen this semester, the genetic material of prokaryotic and eukaryotic cells is DNA, and they use the DNA to synthesize an mRNA molecule to be translated by their ribosomes into proteins. Viruses, as we will see, are more diverse in their genomes. All cells contain both DNA and RNA. Viruses, however, utilize either DNA or RNA as their genome, but not both. The genomes of viruses are typically much smaller than those of their host, allowing them to package them into their capsids. Viral genomes contain only those genes necessary for viral components. To give you an idea of how small viral genomes may be in comparison to their host, let's compare the human genome with a virus. The human genome contains between 30,000 to 40,000 genes, while the hepatitis B virus has only 4 genes. Why such a gap in size? The host cells must contain all of the genes to synthesize cellular structures and the necessary enzymes for metabolism. Viruses only bring with them the genetic code required to synthesize viral proteins because they utilize the host structures and metabolic enzymes. We also learned in Chapter 7 that the DNA exists as a double-stranded molecule and RNA is a single-stranded molecule. Many viruses will contain double-stranded DNA or single-stranded RNA. However, others may utilize single-stranded DNA or double-stranded RNA as their genomes. The genomes of viruses may exist as linear, circular, or segmented molecules. Segmented meaning that several molecules of nucleic acid are present. All types of organisms may be infected by viruses. Bacteria, fungi, algae, protozoans, plants, and animals alike all have viruses that infect them. Animal viruses and bacteriophages are the most researched of all the viruses. It is easily understood why animal viruses would be of interest to researchers. There are a number of animal viruses capable of causing disease. Research is done to understand the structure and replication processes in order to create immunizations or antiviral treatments for such viruses. Those that infect bacteria are known as bacteriophages. Bacteriophages have been researched extensively because they are quite easily cultivated in the laboratory. Bacterial cell cultures are grown and the virus is added to the culture to cultivate more bacteriophages. Bacteriophages have allowed scientists to study viral replication cycles. The range of organisms that a virus can infect is known as its host range. Most viruses infect the cells of a particular host or species. This selection of a virus for a particular host cell is based on the interaction between proteins on the surface of the virus and receptors on the surface of the host cell. For the virus to be able to infect a host cell, the cell must have a receptor that binds to the proteins of the virus. In some cases, the viral surface may be able to bind with a receptor located on the surface of cells found in multiple species, such as the West Nile virus, which infects birds, some mammals, and some reptiles, or rabies, which can infect a number of species of mammals. Such viruses are considered generalist. Other viruses, such as the human immunodeficiency virus, may only be able to infect a particular cell type within a specific species. Again, remember that the ability of a virus to infect a host is dependent on the interaction of viral surface proteins with host cell receptors. Most viruses are so small they cannot be observed using light microscopy. As previously stated, the viruses remained a mystery until the late 1800s. The first virus to be studied was the tobacco mosaic virus. The scientists Dmitry Ivanovsky and Martinus Biernik filtered sap of infected tobacco plants through a porcelain filter that would trap bacteria. They demonstrated that the filtrate remained infectious and could cause disease when introduced to healthy tobacco plants. Their experiments demonstrated the existence of an infectious entity smaller than bacteria. They also found that the infectious agent could not be incubated and grown in broth, but would only grow in its host. In 1898, the first animal virus was discovered. Loeffler and Frosch identified that the causative agent of foot and mouth disease of cattle was a virus. In 1901, the first human virus, the yellow fever virus, was identified by Walter Reed. Until the 1930s, scientists were unable to visualize viruses. They only knew that they were small enough to pass through their 0.2 micron filters and that the agent was only capable of replication when in the host. The electron microscope was developed in the 1930s, allowing scientists to visualize the viruses for the first time. The figure shows a representation of the sizes of various viruses. Some of the smallest viruses are approximately 10 nanometers in diameter, and the largest, according to your textbook, is approximately 400 nanometers in diameter. However, scientists have discovered a virus known as the Nimi virus that can be visualized with the light microscope and may range in size to be 700 to 800 nanometers in diameter. Notice in the figure that some of the smaller viruses are similar in size to cellular structures like the ribosomes.